All right, today is my favorite lecture to give because I'm uniquely cynical about all of this stuff. So you guys will get snarky comments from me throughout the whole thing. But it's the overview of microbiomes, and there's way too much to cover here for one lecture. But I'm going to give you the pretty high level overview of where we are and kind of where we're going with microbiome research and how we do it. So first question though, what is a microbiome? Anybody? How do you define a microbiome? Yeah. Yeah? Yeah, so it is largely, so it's contextualized by the environment you're talking about. So you can talk about the human microbiome in general. You can talk about the human gut microbiome. You can talk about, the panda microbiome. Typically, it's dependent on the environment you're actually defining it in, though. So you can sample all of the different microbes in that environment using methods like I talked to you about last class, the sequencing-based methods, or culturing. But the key is to try to identify all of the different microbes within a given environment. So all the microbes within a particular environment, and then you can kind of extend it a little bit to talk about all of their associated genetic elements and genomes. And so some people talk about it that way, some people don't. Largely, it's the important part here is that you define it based off of a particular environment, though. So the key thing here, you guys there's a certain trillion number of cells in your own body. So I have like a trillion Dave cells in me that work together. At some point they might not, and then that's when I don't exist anymore. But right now they're all working together. We're all a cohesive unit. Depending on certain excretory processes, there's either more microbes inside of me than my cells or about the same amount as my cells. So usually, though, there's more microbes inside of all of you in number of cells than there are actually number of human cells inside of you. And so when you count up everything, you're more microbe than human, again, given certain hand-waving back of the envelope calculations. This also means, so the human genome has something like 20,000 genes in it, maybe 18, 19,000 genes. There's at least 50, 60, 70, 100,000 different microbial genes that are inside of you, inside the microbes living inside you. So it's a vast, greater number of diversity in terms of genetic elements than human genes that are actually there, although we can talk about splicing differences and whatnot. There's a couple different ways that we study microbiomes, and this gets into, you'll see microbiome research, there's a lot of funding in it right now, there's a lot of scientific money, there's a lot of industrial money, there's a lot of effort to try to understand how microbiome changes matter in terms of human health and then animal health and all sorts of other things. There's a couple different large-scale ways to actually measure microbiomes and do it in a controlled experimental way. The two main systems that really started this out were with mice and then zebrafish. The picture I'm showing here is basically a sterile chamber. So what you can do with mice is feed a mama mouse a bunch of antibiotics, get her, kill as many microbes as possible inside of her, then deliver the mouse pups by C-section. Stick those pups because they're born sterile. They have no microbes on them if they're born by C-section. Then you can stick those pups inside of these chambers where it's kind of like filters the air in and protects those pups from the outside environment. So if you take those C-section pups, stick them in this chamber, they grow up without microbes in them. And so a lot of the research that you'll see in papers now is comparing mouse or some other animal grown up in chambers without microbes versus ones that are conventionally reared. So when you see this, typically it's called, so if you get all the microbes away from something, you can call it an axenic mouse. But when we talk about them in these chambers, we typically call them gnotobiotic animals. Gnoto means known, biotic means biotic, life. So mouse without microbes are gnotobiotic because we know that they don't have any microbes, so that's why it's known microbes. But As long as you can replace the microbes inside these animals with something that you know is going in, those are technically also notobiotic. But you'll see notobiotic animals, and then we typically compare notobiotic animals versus conventionally reared animals, which are just, mice typically get their microbiome from birth, and then they eat the poop in the cage, and that's how they get it too. So those are conventionally, actually, mice end up being easy to work with because to change their microbiome, you just give them poop from a different mouse, and they tend to eat it and get colonized by it. So those are mice. The other system that really started a lot of the microbiome research was zebrafish. Zebrafish are wonderful little animals that are models for a lot of different human diseases, and they're translucent when they're baby fish. And so you can actually check a lot of the processes just by looking in the embryo and see things like gut movement and colonization of microbes and a bunch of different things. Zebrafish end up being a lot easier to make notobiotic than mice because the zebrafish are fertilized externally, so they make eggs, and you fertilize the eggs. But these eggs inside of them they're sterile. They have no microbes inside them. The microbes are typically on the outside, and then the fish get colonized as they break through the eggshell and swim out into the tank. But if you take these eggs and fertilized eggs and hit them with a bleach and some antibiotics, you can kill all the microbes on the outside, and then if you stick them in sterile water and sterile filters, filtered water, you can grow up notobiotic zebrafish and then do things like add back different microbes to them. So they're a little bit easier to work with, and you can actually see the developing embryo and see how microbes might affect gut movement or things like that. So these are a couple key systems for microbiomes. There's a bunch more now. Pretty much every animal or pretty much everything people are trying to look at how the microbiome affects its behavior or development or something now. So these are the first two that were really got a lot of play, but there's a ton more now of different research. The reason I focus though on the fish and the mice is that really early on there was a really cool experiment showing largely that the animals themselves and the type of animal, it can actually determine its microbiome to some extent. So what you can do is you can take notobiotic zebrafish, you can take notobiotic mice, and then give them microbiomes from the other animal. And so when you do that, what you end up seeing is that in the controls here, you just take notobiotic mice and give them conventional mice microbiomes, and they look like a mouse microbiome. Same thing with zebrafish. If you take notobiotic zebrafish, give it zebrafish microbiomes, they look like zebrafish microbiomes. If you give the zebrafish a mouse microbiome or give the mouse a zebrafish microbiome, the microbiomes are different enough where you can tell zebrafish from mouse. The host will actually select for not necessarily the exact strains, but it selects for the configuration of the microbiome that matches the conventional formation of it. So if you give a zebrafish a mouse microbiome, the zebrafish can select for changes in that microbiome that more match the conventional zebrafish microbiome and vice versa. So there is some host selection in microbiomes. Your microbiome, human microbiomes, are going to be slightly different than chimpanzees. They're going to be slightly different than lemurs. They're going to be more different than zebrafish and mice, although there is, within vertebrates, there's a lot of overlap in terms of the microbiomes. Phylogeny and genetic history ends up being not the greatest determination of microbiome variance from one individual to another. But there is some selection by your own immune system that determines what strains can actually live inside of you. Perhaps the greatest influence on any microbiome is going to be what you eat, at least in terms of the microbiome of your gut. So a lot of these microbiome studies that you'll see in the news or in other papers, typically the microbiomes are measured by poop. So we sample whatever microbes are in feces of whatever animal we're looking at, because that's the easiest way to sample, and it somewhat reflects the microbiome that's going on in your GI tract. Your arm microbiome is going to be different than your poop microbiome, but we kind of just assume that they're correlated in some degree. But in terms of microbiome in general, like I said, so host determines it a little bit. So mouse is different than zebrafish, but within mice there's a lot of variation. Within humans there's a lot of variation. So person to person, your microbiomes are going to be fairly different, although within the broad spectrum of they all kind of look like a human microbiome in some way. The main determinant of your own microbiome configuration is going to be your diet or your environment. So if we're talking about gut microbiomes, the diet is going to be what truly determines what your microbiome looks like. And this actually plays out across all animals too. So researcher Ruth Lay went to a bunch of zoos. This is actually a pretty cool study that happened early on too. So Ruth went to, I think she was in St. Louis at the time. St. Louis has a pretty good zoo, so she could go there and just collect poop from a bunch of different animals and then match that up with what they ate. And then she got some feces from other places, some other zoos as well. But when you do this, these are all vertebrates, so they're all fuzzy things that we like to look at in zoos. And then what this graph is actually showing is this is called the PCA analysis. So it's breaking down variation in the microbiome based off of two effectively made up axes, but it separates the variation. The key point here is that each of these dots is colored by the diet of the host that it came from. So the blue dots are omnivores, the green dots are herbivores, and then the red dots are carnivores. What this is showing is that the variation in what kinds of species are present within each host kind of groups with each other. So all the blue dots kind of group together to the exclusion of the red dots, to the exclusion of the green dots. And that kind of means regardless of what host we're looking at, the herbivores kind of group together in terms of what microbes live in their gut, the carnivores group together in terms of what microbes live in their gut, and then the omnivores kind of group together. So again, there is a signal from history and phylogeny and your own immune system, but really for gut microbiomes, the diet is going to determine largely what they look like. There's this one little circle down here, so pandas and bears. What do pandas eat? But that's what everybody tells you, right? Pandas end up being like horrible, murderous creatures. Like if you stick a chicken in front of a panda, it'll rip it apart. So pandas end up being weird because and bears are kind of omnivores in general too. So pandas, we kind of classify them as herbivores, but really they'll eat birds if they land in front of them. So anyway, they end up being down. Bears are just weird in general because they eat a bunch of different things, and pandas are in here because pandas are mean. Yeah. But largely though, and what I'm showing you up here is that, I think this is mouse and human. So largely though, the microbiomes within the variation depending on the diet, the microbiomes of vertebrates largely look alike in terms of what constituent microbes are living there. So this is just a phylogeny of mouse and human microbes in blue, mouse microbes in red. All I want you to get from this, from the narrative here, is that the blue and the red kind of match each other. And that just means that in terms of the histories of the microbes, the species of microbes that are living inside mice and humans, they are very similar. So there's a lot of overlap between blue and red, which means that overall, fairly similar microbiomes from humans to mice within the variation of things like diet and stuff like that. So your microbiome is going to be different than mice, but not as different as something like a plant would be from anything else. But again, there is this signal where microbiomes kind of recapitulate the history. So you're going to have a human microbiome that's going to be slightly different than the chimpanzee microbiome, slightly different than the gorilla one, and more different than the mouse. So they're diverging in some way that we really don't have a good handle on how these microbiomes diverge yet. But they are diverging. And so there is some signal there. It's early days, still even early days, to try to figure out what's actually determining these things. This is all complicated by the fact that when we talk about microbiome studies, largely we're talking about those gut microbiomes because that's been the easiest thing to sample across animals and across everything else. And it's not even the gut microbiome. Again, it's the microbiome of whatever makes it out of the gut and is able to be sampled very easily. When you look at human bodies, though, we have a variety of different environments on us. So your armpit's going to be very different than your arm just because of things like humidity, pH, temperature, all sorts of different environmental variables, and that's going to change what kinds of species live in each environment. So, your microbiome is going to be different in subtly different ways throughout your body, and even that variation is hard to compare from one animal to another. But there's a lot of variation that goes on here, and some people are going to, depending on if you're out in the sun a lot, if you're not, if you've got eczema, if you don't, all these things kind of factor into what your microbiome looks like. So diversity within humans and within all animals really is high within your body, and then between species it's pretty high as well. So anytime you see a lot of these microbiome studies, there's always like one clear result or one result that they highlight. There's a lot of variation out there that's changing all the time and can be changed all the time. Questions so far? Okay, now we get into the good stuff. So what do you do as a researcher? If you guys end up being researchers one day, It's very tempting, and you have kids, it's very tempting to actually do research on your own kids if they're there. So this is actually a graph. I think these are two people at Stanford, and they had a baby, and then there were microbiome researchers, so they're like, let's just sample our baby and see what the microbiome looks like over time. There's all sorts of other ethical agreements that you have to get. If it's your own kid, like, the ethics are a little bit weirder than if it's somebody else's kid you're doing this on, so it's a little bit easier. So what they did was this woman had a kid, and then they sampled the microbiome at a variety of different days. The microbiome in humans changes, and it changes in a way that's related to diet, and this is the gut microbiome. If you have a kid, you will know that it's very easy to sample the gut microbiome, very easy some days, because you're up to your arms in diapers a lot of the time. The human microbiome changes though, and so what I'm showing you here is that your own microbiome, there's a lot of diversity and a lot of changes that happen in the early stages of when you're born, and then develop. As you get older, it begins to stabilize though. So what these researchers found, and this is kind of borne out in a few other studies that have been looked at now, like this is the same trend that you see. So these bars represent, so imagine that this whole length of the bar is 100% of the microbiome, and then each of the colors within the bar represent different percentages of microbes that are present within this kid's poop. So here, when it starts off at 3 days, the kid is only eating breast milk. It's pretty much firmicutes for that 3 days. 4, 5, same thing. 6, you get a little bit of proteobacteria that show up, but then they go away days 10 and 14. So early on, largely firmicutes in this kid's gut. And I think they were, this kid was conventionally born, so born conventionally born, so not C-section, like vaginal birth. I think so. A lot of your own microbes early on, if you're conventionally born, you're colonized as you come out your mom's birth canal, basically. It's birth. Having seen the miracle of birth, it's a very interesting process that I can easily understand how babies are colonized during that. It's something you will never forget. So early on, so we got day 14, it's still firmicutes, and there starts to be more proteobacteria, but largely, it's largely driven by firmicutes and proteobacteria up through these first days. And this is a reflection of the fact of what the baby's actually eating. So the baby's eating breast milk, and breast milk has a little bit of microbes in it, and breast milk has a little bit of, a lot of sugars in it that actually promote the growth of things like firmicutes and lactobacillus in the baby's gut. And so your human breast milk is very good for your own human resident microbes. There's sugars in it that actually the good bacteria inside your gut really like to eat, and so it kind of is called a prebiotic that can foster a certain good gut community that shows up. That's not to say that formula is terrible, but typically what it looks like is breast milk is better for culturing your gut microbes and getting a good gut microbiome showing up. All right, so then what happens? So then inevitably your kid's going to get a fever. So at day 92, the kid gets a fever. And so what is fever? Fever is the human body's reaction to some foreign infection. Your temperature goes up. If the temperature goes up, the microbes inside of you are really good at growing at like 37 degrees C. If you have a fever that's a little bit higher temperature, that can shift the microbiome a little bit. So here you can see this. So at day 92, this microbiome, after the fever shifts, you get a little bit more proteobacteria, and then some actinos show up. But then they go away after a couple days after that fever. So that brief fever, that brief infection is enough to shift the microbiome a little bit, even in this infant that's just eating breast milk. All right, so they survive the fever. It goes back largely to what it was before, although there's still these actinos that are showing up a little bit more. But largely, day 128 is very similar to day 6 over here. So it's a pretty stable microbiome because they're eating the same things. All right, I will also attest to this. After a while, it gets very difficult to keep feeding your kid breast milk every day. So you want to give them something else. And so at day 134, they started rice cereal. And then they start formula and other table foods and stuff. So you can see a dramatic shift. Not necessarily after the rice cereal, but after they start formula and the other foods, you get a dramatic shift in microbes where these bacteroidetes show up and then kind of dominate from then on. So again, the human diet is factoring in here. It can change everything. And really, a diet is going to really drive what microbes are present inside of you. And again, as we talked about things like 16S ribosomal RNA sequencing, there is diversity hidden in these sequences because the 16S is going to be the same across some firmicutes. And as we talked about with the E. coli example, that can be very different even with the same 16S. But nonetheless, this gives you a reflection of how things are changing. One other thing I want to point out here is that breast milk and formula ends right here. But they had a couple of treatments of antibiotics, so amoxicillin right here, and then cefaldir right here. And so antibiotics are also going to affect your microbiome because that's what they do. Antibiotics are there to kill some pathogenic microbe that's wreaking harm. If you're on antibiotics, you're on them for a reason largely. And so antibiotics are going to both affect the bad microbes that you're trying to get rid of, but they can also affect the good microbes. And as you can see here, with cefaldir, the bacteroides go away for a couple days while they're on the antibiotics, and then they come back after the fact a little bit later. So antibiotics can also shift your microbiome, and there's all sorts of discussions we can have about that. All right, so this was an N of one baby a few years ago. This has been repeated basically the same story shows up. So infant microbiomes are colonized at birth by a variety of different ways. So the C-section microbiomes will be slightly different than conventionally raised microbiomes, just because if there's a baby C-section, there's just, I think in the early stages, C-section babies, their gut microbiome ends up looking more like the skin microbiome than the conventionally birthed baby gut microbiomes. It was actually, so I had a son last year, born 2.5 months early. And the NICU is a weird place because it's every day to actually go in there, you have to scrub down with all these antibiotic washes and stuff. And he was born by C-section too. So you know, my kid started off as a C-section, and then he was in this largely very sterile place, or they try to make it so because they're trying to prevent all these other microbes coming in from the outside world. And so, NICU babies by C-section are going to be different than conventionally raised babies, and we right now, as of right now, we don't have a good answer for whether that matters or not in the long term. In the short term, at least in terms of microbe constituencies, it does differ, but we don't know what the long-term effects of that might actually be. There might be things like asthma or allergies that might be different, but it's early days in figuring out those trends with the microbiome. There's another study though. So this was on a baby over a year or a couple years. A couple years ago, another study was published. This was actually a grad student, so he had to call himself Subject A, but it's his own poop and saliva. So he was at Duke. He was sampling, and what he did was he sampled his variety of different parts of his microbiome over the whole year. And so this is not a baby. This is an adult, like a 20-some-odd-year-old person. So what I'm showing you here is this on the x-axis here is the days, and these gray bars are the days that he sampled his microbiome. These colored bars are the constituencies, just like in that bar graph before, of the different microbes that are going through time. So what I want you to take away from this is, as you're an adult and you're probably eating very similar things, your environment's kind of set up and you're not changing it too much over time. And in humans, we're pretty variable in what we do. So there's kind of an on-average environment that you end up getting. His saliva microbiome was pretty stable over a year. So your own microbiomes, they're not changing very much unless something dramatic happens. So baby microbiomes, if they're changing diets and stuff, pretty stable after a while. Human, adult microbiome, same thing, unless if he goes vegetarian or something, that might change it. However, so he was sampling his saliva, he sampled his own poop, and his gut microbiome. And so there was a dramatic change in one time point for his gut microbiome. And so I'm showing this to you for a variety of different reasons. So overall, if you look at the whole year, day 0 and day 365, the microbiome constituencies look very similar. But day 100 here, this grad student ended up he was in Bangkok for a few weeks, ate some bad food, had some gastrointestinal issues, and food poisoning. And yes, he was in Bangkok, and he had a fridge there storing his samples. His dedicated grad student. But he was there sampling his gut every day, and he had food poisoning. So don't think about the details of it, but the data that comes out of this is that he had food poisoning, and the food poisoning changed his microbiome around day 100. So for a few days here, it was vastly different than when it started off. But after he got over it, it kind of went back to what it was before. So even with those brief disturbances, if you're eating about the same things, largely it stays pretty stable, it looks like, over time, unless you get these brief disturbances for very noticeable reasons. This doesn't mean that within that stability there's not changes, and so your own microbiomes are going to be influenced by a variety of different things. So overall, very stable in terms of 16S taxa that are there, but that doesn't mean that the specific strains and the specific species aren't changing over time, and changing by a variety of different ways. And so, like I mentioned before, your microbiome, if you sample densely enough, you would see that your microbiome is more similar to the person sitting next to you than the person sitting in the back of the room, just by the fact that you're all shedding microbes and you're all kind of exchanging microbes right now. This is one of the good demonstrations of this is you can go, it's another pretty interesting study where they set it up. So this study was run by a team at the University of Oregon. The PI on this study was actually into roller derby, and so she had a roller derby team, and she convinced them to let her sample their microbiomes like before the roller derby match and after the match, and just see. And if you've ever seen roller derby, there's a lot of like skin-to-skin contact and different interactions between people. And so during these roller derby matches, you would see that, yeah, sure enough, different teams would exchange their microbes through that pretty intimate contact, and that was you were able to see that in changes in the microbiome. Even though I have no clue how long a roller derby match is, but it was long enough where you could exchange microbes and actually see that signal of differences in the skin microbiome. It also led to one of the greatest method sections ever, where I've never seen any other paper where booty is an official term, but it's one of the greatest method sections I've ever read. But nonetheless, the intimate contact like that can change your skin microbiome. Another thing that can change your microbiome is the environment around you. And so I live basically on a farm. I've got a bunch of dogs, a bunch of horses, a bunch of other things. It turns out those animals and your cohabitating animals end up shifting your microbiome and increasing the diversity in the microbiome, again, for obvious reasons. If you have a dog, dogs tend to do things like lick you in the face and whatnot. And so that changes your microbiome. So this is a pretty hard graph to digest, but what I just want to show you here is on the right side here are bar graphs reflecting the number of shared bacterial phylotypes, those OTUs, that you can actually go in and sample. And so the blue bar here is, I think, 2 people living in the same house with the same dog. They share a very similar microbiome than 2 people living in the same, so that's a blue bar. The green bar is 2 people living in the same house without a dog. So they share less of the same microbiome. And then this red is 2 houses with dogs and 2 people within those houses, I think. So basically the take home from this is that the blue bar means that if you have a dog, so what you want to compare is the blue to green. If you have a dog, that dog ends up being a vector for microbes between you and whoever else you're cohabitating with. Again, for pretty obvious reasons, because dogs end up getting very up in your face a lot of the time. This is, so depending on who you talk to, they may say, oh, this is great because some changes in the microbiome can be beneficial for things like allergies or respiratory health or other things. And dogs tend to maybe shift the microbiome to make it more diverse in ways that might be beneficial. So there's kind of a push to be like, hey, get a dog because your microbiome might be more healthy. This is a disclaimer where I say that there's no good definition for a healthy microbiome versus a not healthy one. We know when a disease microbiome looks like, because it's things like if you have salmonella or if you have anthrax or something where there's a known pathogen, like that's a noticeable disease microbiome. Anything aside from that, you're kind of arguing about things we don't really understand very well because we can't define truly what a healthy microbiome looks like. So this means that if you see late night commercials that say, send us your microbiome samples and we'll give you a probiotic that is tailored to your personal, complete BS. Like, it's just snake oil right now at the level of like they're just trying to get your money because microbiome research is hot. So, so don't spend money on giving companies, don't give your money to companies for them to give you back personalized microbiomes because we don't understand enough to actually do that right now. Just sampling the microbiome is one thing. Developing them for personalized medicine, completely different questions right now. Incidentally, also don't give money to places that will give you your telomere age because that's kind of BS as well. they're just doing PCR on your telomere ends, and they tell you, oh, your telomeres are 84, but you were 35. Like, okay, thanks. Like, that's great. Anyway, it's a $100 test, and it's completely, there's nothing from it that you can really tell. Anyway, same thing with personalized microbiomes. I'm going to come back to, so you guys want to know the Dannon, the probiotic yogurt answer? So probiotic yogurt, so all yogurt has microbes in it. That's kind of what makes it yogurt. It has lactobacillus, it has streptococcus. Yogurt has good microbes in it. Dannon Activia is a little bit more expensive than other yogurts, and there's other probiotic yogurts out there. It ends up being there's no clinical benefit from Dannon Activia versus any other yogurt, because if you're eating yogurt, you're eating microbes anyway. It even is worse than that, because the domesticated, there's bifidobacterium that they call it, that's in Activia, that makes it kind of a probiotic yogurt. The bifidobacterium that is trademarked for and patented by Dannon has been passaged in the lab enough where it actually doesn't really colonize your stomach at all. Like if you eat a yogurt, if you eat a yogurt for like 5 days straight and then stop eating yogurt, the bifidobacter is gone like 3 days later. Like you won't see any trace of it. The lactobacilli really don't colonize your gut very well at all. The benefits of yogurt are largely because you're eating yogurt and there's microbes in it anyway. So it's all marketing. Yogurt's good to eat. It helps out your stomach, but the probiotic yogurts there's no clinical evidence that they actually do anything compared to regular yogurt out there. So yeah, there's a lot of marketing here that they try to get you with this. Plus the probiotics, it has to go through your stomach, and your stomach kills a lot of stuff when it goes through it. So it's, yeah, there's a lot of companies out there trying to get your money. But eat regular yogurt, great. Like just don't spend the money on the extra probiotic yet. I'm not endorsed by Dannon, so. There are a couple cool stories though where there does seem to be some more long-term evolutionary relationships where human microbiome has been configured in a way that might allow you to eat certain foods. And so we don't quite know the follow-up to this study yet, but there was an interesting interaction scene where folks in Japan have been eating seaweed for a lot longer than folks anywhere else in the world, things like sushi and whatnot. The human gut microbiome, so your own gut microbiome, contains a variety of different diverse enzymes to utilize polysaccharides. You know, what is seaweed? It's just a lot of polysaccharides put together. If you actually look in the gut microbiomes though, so there's this Bacteroidetes plebeius, so that Bacteroidetes was that yellow bar from the early on baby study when they changed it to table food. Bacteroides ends up being a dominant gut microbiome member for all humans, and it actually helps digest a variety of different foods. Bacteroides plebeius contains enzymes to break down sulfated polysaccharides like ones you might find in seaweed. But genes for these enzymes are only found in Japanese microbiomes so far, not in the North American ones. So in North American microbiomes, we have Bacteroides. They just don't have the genes to break down the exact polysaccharides that are utilized in things like sushi and other seaweed-based foods. So it seems like these might have been acquired by horizontal transfer by these species, because if they're living in the intestinal tract of people that eat seaweed for a long time, it makes sense that if you're a microbe that makes your living off of breaking down whatever food comes through the GI tract, if this food is largely consisting of a variety of different seaweed-based things, you know, that it's an open source there for the bacteria to actually learn how to break down those polysaccharides. So again, it seems like this result holds up, and there's some other cool findings like this that kind of go along with it. So it seems like your gut microbiome can be changed by the foods you eat in an evolutionarily relevant way. What this means for personalized microbiomes, no clue yet, but it's just, you know, what we can do is we can sample what's there and then see what the correlations are with that. So why do we care about all this? We really care about all this because microbes influence pretty much all developmental and physiological processes across life at some level. Every eukaryote in existence has lived in the context of microbes. Since you were born, there's been microbes colonizing all throughout you. And there's this hint that the type of microbes that you're colonized by might affect larger health trends. It's pretty obvious when we're talking about very acute sicknesses like anthrax, or if your microbiome contains anthrax, you're probably not doing so well. Same thing with things like Shigella, Salmonella. Those acute diseases, it's pretty obvious that the microbiome can matter. But then the question becomes, what about the other more complex diseases, the ones where there's not an acute treatment, maybe the chronic ones, maybe some other instances? How much does the microbiome matter there? And the challenge with this is that basically any disease that involves inflammation, so inflammation is your body's defense against infection. So inflammation in itself kind of affects the microbiome, and so there's a lot of diseases out there that affect inflammation pathways that then therefore affect microbiomes. And we have arguments with people all the time about whether things are correlation or causation in terms of whether the microbiome's driving inflammation or whether it's just the inflammation kind of driving the microbiome. But you'll see that in a variety of different ways. So one of the current ones now that is still hugely up in the air, and I have no clue where it's come down, but so Alzheimer's disease ends up being a disease of inflammation. So if you look in people with Alzheimer's, there's a microbiome correlation to Alzheimer's and potentially even in the brain. So you get some people wanting to say that Alzheimer's is caused by microbes or fungi. Hard for me to believe that that's true, but that's the correlation out there now. And there's some people trying to say that, you know, there's that lure of like making your career finding, and then you kind of go with that for it. So you'll see things like that where there's diseases of inflammation that where people are proposing microbiome influences of it. And it's still kind of unclear now as to what's actually going on there. But microbiomes do matter at some level for things like plants. So plants, these guys on the left are not inoculated with a plant growth promoting bacterium. These ones on the right are. So you can put certain strains on plants to make them grow better. These probiotic strains end up being ones that do things like make hormones or provide limiting nutrients to the plants. There is a definite trend where the microbiome inside of your gut affects your obesity. However, people want to, so I love this fat mouse, skinny mouse picture. This is actually not from a microbiome paper. It's from a different, they're genetically different mice, but I always show this because it makes the case. So there is a microbiome associated with obesity, and there's a microbiome associated with the opposite of obesity, which I'm blanking on what the word is right now for that, but the skinny mice and fat mice have different microbiomes. There's a small effect where the microbiome drives that, but really it's because some microbes tend to be able to break down and get calories out of different kinds of food than other microbes, and if you're tending towards the obese side, you tend to be over biased towards those microbes that can eat and break down those different foods. Small effect size of whether the microbes make you more obese or skinnier, but it's not going to be like a 20, 40, 60, 100-pound thing. It's like, you know, a couple pounds here or there can be attributed to what microbes are inside you, breaking down the different foods. Significant difference, but not, you know, you're not going to be able to get a probiotic and make you fat or skinny. It's much more complicated than that, but there is an effect size there. In chronic disease, like obesity, Crohn's, allergies, cancer, we talked about Helicobacter. There's evidence that the microbiome matters for these chronic diseases, although the evidence is, the argument you have to worry about there is how much of the disease actually affects it, or is affected by it. So microbes won't give you diabetes, but they can make diabetes worse. Microbes won't make you fat or skinny, but they can make those two things more exacerbated. It looks like microbes might be involved in Crohn's disease, but it's more complicated than that because there's feedbacks with your own immune system and the microbes there. Allergies, sometimes it looks like there's a microbiome effect there, still kind of debated. Cancer, there's at least one microbe, that Helicobacter pylori, that is involved in cancer. There's some other ones that might be, although because cancer is a disease of inflammation at some levels, there's actually a cool thing where for things like colon cancer, you can screen the microbiome of your colon, and there's a subtle change that happens in the microbiome that's diagnostic of colon cancer before you can actually see the colon cancer showing up on other tests. So we might be able to do things like use changes in the microbiome as a reflection of larger changes in your own health. And then there's all sorts of other things like probiotics. Here's my 2-minute take on probiotics. If you're going to take a probiotic, I showed you before that graph of the grad student's microbiome where it's largely stable over time. Your microbiomes are going to be largely stable over time, depending on, as long as you eat the same things here and there. If you take a probiotic, it might make a small difference, but that strain is probably not going to be able to outcompete whoever else is there in your gut. It's not going to take, probably. And so a lot of the effects might be kind of placebo things going on. But that's not to say that there's no effect there. There's depending on what other microbes are there. If you've had large changes in your microbiome from eating bad food in Bangkok, probiotics might help to help recolonize and get things back to normal after the fact. But probiotics largely are, again, it has to make it through your stomach, and a lot of things die in your stomach. So the easier way to change your microbiome, if you're worried about microbiome being bad or good or healthy or not healthy, there's what are called prebiotics. So prebiotics are basically food that can nourish your microbiome in a certain way. That ends up being a lot easier way to change your microbiome because the food is digested and then makes the food doesn't have to be killed in your stomach. Or you can change your diet or whatever, and that'll change your microbiome if you're worried about it. So the easier way, if you're worried about your microbiome, to change it is to change your diet or change your other behaviors, because that's probably going to have a larger effect than taking pills to switch it, unless the pills are antibiotics, in which case that's a whole different story. Some effects of microbiome on correlated diseases. So IBD, there's a microbiome change. Again, it's inflammatory, so there's inflammation that's going to change the microbiome because inflammation is a defense response. Type 2 diabetes, again, this gets into the obesity versus non-obesity argument. The microbiome can make it worse. It's not going to give you diabetes, though. There's other correlated things there that are helping out. In infants, so necrotizing enterocolitis, this is the one where there is a pretty good evidence of microbiome mattering. So necrotizing enterocolitis is in NICU babies, it's horrible. Basically, you can read what necrotizing. Anytime you see the word necrotizing, it's not good. Yeah, so the microbiome matters here for necrotizing enterocolitis. There's certain strains that can colonize babies' guts to prevent other strains from infecting them. In NICU babies, again, the NICU ends up being a weird place where the microbiome is set up in a different way than it's conventionally there. And so you can get these bad diseases that show up because of changes in the microbiome. The one thing I'm going to go through in terms of more details today is how the microbiome can change with these invasive pathogens, so things like salmonella. So if you just take a mouse with a normal microbiome and give it salmonella, the mouse isn't going to get salmonella very easily. You have to give it a ton of salmonella for it to matter. So largely, and your body works the same way. If you eat a piece of chicken, if there's a little bit of salmonella in the chicken, you're probably not going to get salmonella. To actually get salmonella to work in the lab in the mouse model, we need to give the mice streptomycin, which takes out their gut microbes. It kills a lot of the gut microbes and changes their microbiome. And then we can actually give the mouse and infect it with salmonella. So what's actually going on here? In your gut, there's the bacteria and food and the intestinal lining and then your gut lining. Imagine here on the right is the intestinal lumen. Here, these blue guys are just normal gut microflora that are in your gut, hanging out with the lumen. What salmonella ends up doing, so these yellow guys, imagine those as salmonella. So what salmonella ends up doing as an infection strategy is it'll infect, but if these normal other things are there, they pretty much colonize the niche that salmonella would colonize. And so your normal gut resident microbiota protects pretty well against salmonella. But what salmonella can do is salmonella has a type 3 secretion system that can infect this intestinal lumen, and this infection and the proteins that are translocated by the type 3 system lead to more inflammation in the gut. And so that inflammation in the gut, again, inflammation is going to change your microbiome. That inflammation ends up killing a lot of these normal gut microbes and opening up the niches for salmonella to actually populate your gut. And then that's basically how you get salmonella diarrhea, is because all the regular microbes that would normally defend you are gone due to a type 3 secretion system. And then salmonella overtakes everything. The other disease you'll see with microbiomes where there's a really clear trend where microbiomes matter is with C. diff, so Clostridium difficile. You'll see this. It's a major cause of diarrhea in hospitals. It's a major cause of death and other problems after surgeries. Because what do you do for surgeries? Well, your doctors give you a high dose of antibiotics to try to clear out any infections and protect your health. These antibiotics, these broad spectrum antibiotics, are going to target everything, though. So they target the bad guys, they target the good guys, and if you target your good microbes, it gives C. diff a niche that it can actually. So C. diff ends up being really resistant to a variety of different antibiotics. So C. diff isn't killed by the broad spectrum antibiotics, but your normal good microbes are killed by them. So if those normal good ones are killed, then C. diff has a free reign to kind of go and populate your gut. This can lead to death largely through dehydration. So again, it's this inflammation thing where you get a loss of water. So the reason I mentioned C. diff though is that C. diff is one of these like infections where if you get it in a hospital setting, there's some bad outcomes a lot of the time. However, there's a really simple treatment for it that seems to work a lot of the time, and that's fecal transplants. So because C. diff is a disease of the absence of good microbes, allowing for C. diff to actually populate your gut or your colon, if you take fecal transplants from a healthy patient and again put them in the right way, because a lot of the bacteria die in your stomach, so if you put them in the right way, I'm trying to be nice here. So if you transplant fecal bacteria from a healthy person to one with C. diff, a lot of the time the C. diff will be treated very easily. So this has led to all sorts of ethical discussions and weird people trying to do fecal transplants. And yeah, there's people with inflammatory GI tract diseases. It's a horrible condition. They're horrible conditions, and so any hope that you can change your microbes in a good way, there's been some stories of there's like fecal transplant clubs and stuff that people have kind of formed. Nonetheless, it seems like for C. diff at least, fecal transplants end up being a pretty easy cure in many cases. For other inflammatory diseases, not so much. If you drink too much diet soda, it can basically what happens, and this is if you drink diet soda solely for like 2 weeks straight, your gut microbes that can break down glucose end up being biased against, and they go away. And so then if you take a diabetes test, your body is really poor at breaking down glucose, which is the diabetes test. And so because your gut microbes the ones that can break down glucose have been lost. So anyway, if you drink too much diet soda or you eat too much aspartame or saccharin or other things, it can shift your microbiome, and that might have false positives or false negatives for other tests that might be based off of what you expect normal humans to function like. Last thing I want to go over is that these microbiomes, you do have your microbiome is stable, you have a cloud of microbes, you leave that cloud of microbes where you go. So there is a lot of money being spent to figure out, can we go to a crime scene or can we go to a computer or whatever and see who was using that computer or who was at the crime scene based off of their strains of microbes that were there. So it seems like if you sequence the microbes to a high enough depth and get strain-specific precision, you can basically place people in certain places based off of the microbes that are there and the specific mutations in those microbes. You can also use this as so your microbiome shifts. So when you die, all your cells basically start lysing, and that shifts the microbiome. So it seems like the microbiome ends up being a pretty good clock for telling you how long ago you died. There's a whole, if you guys know, so in Tennessee, University of Tennessee has a body farm out in the middle of the forest. So don't ever go backpacking through like the middle of Tennessee, because there's a body farm. So what people do is they get their bodies donated, then throw them out in the forest, and then we just sample the microbiomes over time as the bodies decompose. It's a great place to go. But yeah, so there's studies like that where we're measuring over time how the microbiome changes. This is the summary, but largely don't spend money on fad microbiome things yet, because we don't understand it that well. See you guys on Monday.